number 2. I'm Sophia. I'm the Vice President of Academics for LearningLink. This article is pretty short. It's only about 2 pages, but there's a lot that we can talk about. So low commitment, but lots to talk about during journal club, and I look forward to it. I mostly just wanted to break down the title and basically just the whole basis of the experiment through that. First of all, what is a double dissociation? So this is a research method. It's not really a methodology, it's just kind of like a certain set of results that you can get from your experiment. So a double dissociation is when you compare 2 patients with different brain lesions, and a lesion is when there's physical damage to a brain area, or basically it just isn't functioning properly. So we can also have virtual lesions or temporary lesions by messing with the electrical activity of neurons. But here, these patients have permanent lesions, and we'll get into that later. So basically you're comparing 2 different patients with 2 different brain lesions, and you compare them in 2 different tasks. And one will have, as you can see in these graphs, one patient will have a deficit in task A, but do fine in task B, whereas the other one will be fine in task A and have a deficit in task B. And the significance of double dissociation is that it proves independence of the brain systems responsible for each task, whereas a single dissociation, where you have a lesion in one brain area and the patient performs with a deficit in task A, but not a deficit in task B, that only proves that the area lesioned is necessary for task A. It does not prove that the 2 different tasks are separate brain systems, or that even that this area isn't associated with this function. Maybe other areas are making up for it. There's a whole lot of explanations. And maybe task A was actually way more difficult than task B, and that's why there was a deficit. The difference between asking you to speak the word supercalifragilisticexpialidocious for testing speech production, but then comprehension asking you to understand what egg means. Maybe the tasks weren't at the same difficulty level. So anyway, this is a double dissociation, which is the gold standard, so that's a good thing. Next, I wanted to go over conditioning. This is classical conditioning, which some of you have probably heard of from AP Psychology. It's basically when an organism associates 2 stimuli, which previously that were not associated together. So there's an unconditioned stimulus, which is the stimulus that you automatically have a reaction to without learning. So in this study, there's a loud boat horn, which startles participants. And the conditioned stimulus is the stimulus that gets associated with the unconditioned stimulus. And then once the unconditioned stimulus is removed, you still have that reaction that the unconditioned stimulus produced. So in the study, the loud boat horn gets associated with the blue screen. And the blue screen, even though seeing blue does not startle you, after associating it with the boat horn, you get startled after just seeing blue. That is classical conditioning. And then what is declarative knowledge or declarative memory? It's facts. It's something you can articulate to someone else, recalling an event, which is particularly relevant to the study. This is different from procedural knowledge, which is the muscle memory of like tying your shoes or how you draw something. And it's also different from emotional memory and associating emotions with, I don't know, certain stimuli or scents or events or people, etc. This is like very facts-based. And again, keep that in mind, that emotional memory and declarative knowledge are not the same. That's relevant to the study. So, there are basically 3 groups in the study. One is the controls, normal patients, no brain lesion. And then there's this SM patient who has Urbach-Wiethe disease. And normally Urbach-Wiethe disease is characterized, you think of people with this like bad skin condition, but it also affects you neurologically, and it affects in calcifications forming in blood vessels in the brain. And so this results in different, normally it's calcifications in blood vessels to the medial temporal lobe, but this can affect people different ways. In SM, it was pretty darn localized to the amygdala. As you can see in a control patient, the amygdala is fine, and this is gone, because there were calcifications in blood vessels leading to the amygdala, and it died. So she does not have an amygdala. That is the lesion to SM, is to her amygdala. The other patient is called WC. He has a lesion to his hippocampus as a result of an ischemic stroke. So the double dissociation is between these 2 participants, or these 2 lesions, one to the amygdala and one to the hippocampus. And the study investigates how this affects declarative knowledge, and then classical conditioning, or like emotional associations.